Aerobic respiration. This is the process in which ATP is produced, the universal energy currency of the cell, by the breakdown of glucose in the presence of oxygen. Glycolysis is the first step in which cells produce this essential energy. But how? Glycolysis begins outside the mitochondria in the cytoplasm, with the first 5 steps being the energy investment phase, where glucose is used. Glucose that's taken from the food we eat enters the bloodstream, when glycoproteins, also known as glucose transporters, carry glucose into the cell to the cytoplasm. The enzyme hexokinase acts as a catalyst for this next reaction, when ATP is reduced to ADP and an inorganic phosphate. The inorganic phosphate attaches to the glucose molecule, creating glucose 6-phosphate. This traps the glucose inside the cell, preventing the bidirectional glycoproteins from taking them right out. From glucose 6-phosphate, the enzyme phosphoglucomutase catalyzes an isomerization reaction, rearranging glucose 6-phosphate into its isomer, fructose 6-phosphate. An isomer is a compound that has the same chemical formula but different arrangement of atoms. This rearrangement gives the atoms different physical and chemical properties. Third step involves the use of another ATP. This is another phosphorylation reaction, where the enzyme phosphofructokinase is used. ATP is reduced to ADP and an inorganic phosphate. The phosphate attaches to the first carbon molecule, creating fructose 1,6-bisphosphate. Through the enzyme aldolase, a lysis reaction occurs, which is the breakdown of a molecule into smaller parts. In this reaction, fructose 1,6-bisphosphate splits into dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P, both 3-carbon molecules with a phosphate attached. DHAP, dihydroxyacetone phosphate, then undergoes an isomerization reaction, where it gets converted to an additional glyceraldehyde 3-phosphate through the enzyme triose phosphate isomerase. This happens because only G3P can continue down the glycotic pathway. It's important to note that from this point on, everything happens twice. This is because there is now 2 G3P molecules. We now enter the energy payoff phase. The G3P then gets converted to 1,3-bisphosphoglycerate, catalyzed by the enzyme triose phosphate dehydrogenase. 2 things happen here. First, the aldehyde group of G3P is oxidized, releasing electrons. NAD+ takes these electrons, reducing to NADH. Next, phosphorylation, when an inorganic phosphate gets added to the reaction, resulting in a phosphate on the first carbon. ATP is then directly produced when ADP takes the phosphate from the 1,3-bisphosphoglycerate, resulting in 3-phosphoglycerate through a substrate-level phosphorylation reaction. This is catalyzed by the enzyme phosphoglycerate kinase. 3-phosphoglycerate then undergoes a mutase reaction, where the phosphate group shifts from the 3rd carbon to the 2nd carbon, creating 2-phosphoglycerate. This is catalyzed by the enzyme phosphoglucomutase. 2-phosphoglycerate then undergoes a dehydration reaction, catalyzed by the enzyme enolase. Enolase removes water from 2-phosphoglycerate, resulting in phosphoenolpyruvate, or PEP, a high-energy molecule. The last step of glycolysis converts PEP into pyruvate. This is a substrate-level phosphorylation reaction, where the high-energy phosphate group is transferred to ADP, directly forming ATP. This is catalyzed by the enzyme pyruvate kinase. At the end of glycolysis, we made a total of 4 ATP and used 2, leaving us with net 2 ATP. We produced 2 NADHs and 2 water molecules. Pyruvate oxidation is the next step of aerobic respiration. This process connects glycolysis to the Krebs cycle. Pyruvate oxidation occurs in the mitochondrial matrix. This process converts pyruvate into acetyl-CoA through the enzyme pyruvate dehydrogenase. Pyruvate is first transported into the mitochondria and then undergoes a decarboxylation reaction, where carbon is removed in the form of CO2. This forms an acetyl group, a 2-carbon molecule. The 2-carbon molecule then gets oxidized, and NAD+ is reduced to NADH by capturing the high-energy electrons released from the oxidation of pyruvate. The acetyl group is then attached to coenzyme A, forming acetyl-CoA. At the end of pyruvate oxidation, we created 2 acetyl-CoAs, 2 NADHs, and 2 CO2s, carbon dioxide, that was released. The Krebs cycle, also known as the citric acid cycle, is an 8-step process that also takes place within the mitochondrial matrix. This cycle fully oxidizes acetyl-CoA. The Krebs cycle is responsible for producing NADH and FADH2, high-energy electron carriers that fuel the electron transport chain, which is crucial for ATP production. Acetyl-CoA, a 2-carbon molecule, combines with oxaloacetate, a 4-carbon, to form citrate. This is a condensation reaction, catalyzed by the enzyme citrate synthase. Citrate is then rearranged to form isocitrate, a 6-carbon, through the enzyme aconitase. This is classified as an isomerization reaction. Isocitrate gets oxidized through the enzyme isocitrate dehydrogenase to form alpha-ketoglutarate. In this process, a decarboxylation reaction occurs when carbon is lost in the form of CO2, making alpha-ketoglutarate a 5-carbon molecule. NAD+ gets reduced to NADH by accepting the high-energy electrons that were released. Alpha-ketoglutarate, a 5-carbon, gets oxidized to succinyl-CoA, a 4-carbon. This reaction is catalyzed by the enzyme alpha-ketoglutarate dehydrogenase. During this reaction, a decarboxylation occurs, where carbon is lost in the form of CO2. The oxidation of alpha-ketoglutarate releases electrons which are accepted by NAD+, which then gets reduced to NADH. Succinyl-CoA then gets converted to succinate through the enzyme succinyl-CoA synthase. Here, GDP combines with an inorganic phosphate, forming GTP. ADP then steals the inorganic phosphate group from GTP to produce ATP through substrate-level phosphorylation. Succinate then gets oxidized to form fumarate through the enzyme succinate dehydrogenase. This is where FAD is reduced to FADH2, an additional electron carrier, when the electrons released through the oxidation of succinate gets picked up by FAD. FAD is reduced rather than NAD+, because the energy released in this reaction isn't high enough to reduce NAD to NADH, but does have enough energy to reduce FAD to FADH2, which requires less energy to accept electrons. Fumarate then gets converted to malate. This reaction is catalyzed by the enzyme fumarase. This is done through a hydration reaction, where water, H2O, is added. Lastly, we have the oxidation of malate. This is done through the enzyme malate dehydrogenase, where malate gets oxidized to form oxaloacetate. Through this reaction, NAD+ gets reduced to NADH. At the end of the Krebs cycle, we made 3 NADHs, 1 FADH2, 1 ATP, and 2 CO2s. However, this is for one turn of the Krebs cycle. In simpler terms, per 1 acetyl-CoA. But remember, we have 2. This means that the Krebs cycle happens twice per glucose, meaning that per 1 glucose molecule, we have 2 turns of the Krebs cycle, making a total of 6 NADHs, 2 FADH2s that are utilized in the electron transport chain, 2 ATPs, that is, usable energy, and 4 CO2s that we exhale as waste. The electron transport chain happens within the inner mitochondrial membrane. Its main goal is to use high-energy electrons from NADH and FADH2 to produce ATP through a concentration gradient. The first step of the electron transport chain occurs when NADH donates its electrons to complex 1. Essentially, NADH gets oxidized to NAD+, and releases a proton and 2 electrons that are transferred into the first complex. The electrons are then passed to ubiquinone, a mobile electron carrier. The release of energy is used to pump protons from the mitochondrial matrix into the intermembrane space, which contributes to the concentration gradient. FADH2, produced in the Krebs cycle, donates its electrons to complex 2, bypassing the first complex. FADH2 contributes less energy than NADH. Complex 2 does not pump protons, but it still transfers electrons to ubiquinone. Ubiquinone, the mobile electron carrier, brings these electrons from both complex 1 and 2 to complex 3. Once the electrons reach complex 3, the electrons are passed to another mobile electron carrier, called cytochrome C. When the electrons are transferred, the release of energy is used to pump protons into the intermembrane space, adding to the concentration gradient. Cytochrome C then transfers these electrons to complex 4. At complex 4, electrons are accepted by the final electron acceptor, oxygen. The oxygen then combines with these electrons and protons to form water, H2O. This is a very important reaction because it allows for the electron transfer chain to continue by clearing the electrons. Complex 4 also pumps protons from the mitochondrial matrix into the intermembrane space, further contributing to the concentration gradient. The concentration gradient created by the pumping of electrons at complexes 1, 3, and 4 builds up a high concentration of protons in the intermembrane space compared to the matrix. This allows for proton motive force, which stores potential energy that's used to produce ATP. The stored energy gets released as protons flow back into the matrix through an enzyme complex known as ATP synthase. As the protons pass through, they cause a part of the complex to rotate, causing a phosphorylation of ADP plus an inorganic phosphate, creating ATP, through a process called chemosis. Overall, aerobic respiration produces a theoretical yield of 38 ATP, a crucial form of energy that powers essential processes like muscle contraction, nerve impulse transmissions, and chemical synthesis, essentially fueling all cellular activities.